Hi guys, this is a review of Lecture 8 and 10, Male and Female Reproductive Physiology. This material we've covered a number of times. Mostly the histo lectures had it, so hopefully we could just speed right through this, but it's good stuff. Hope you guys are doing well. Alright, so starting off with overview of the male reproductive system, we of course know we have our testes, which are going to produce sperm, and then our testosterone. Then we have on top of it the epididymis, which we can see there. This is going to aid in sperm maturation and transport. Then we have the, continuing on, the ductus deferens, which is going to aid in sperm transport. We said that it had, super thick, smooth muscle around it in 3 layers. Then we get to, our seminal vesicle, the first of our accessory glands. We have our prostate. We get to our ejaculatory ducts, and then our Cowper's gland overall. All right, and then finally we end in our urethra. We're talking about the passage of sperm overall. All right, so going back to the testes, which is where we know spermatogenesis is occurring in particular within our seminiferous tubules and these different lobules we had. Our spermatogenesis is happening in 3 phases, right? We're starting with our spermatogonia, which is on our basal compartment. And so the first of our 3 phases for spermatogenesis is the spermatogonial phase, where we have 3 types of spermatogonial cells. We have type AD, which is type A dark. We have type A pale, aka AP, and then we have type B. And so type AD will divide and make type AP via mitosis, or will make more of type A dark. Overall, type A pale will undergo mitosis to become chains of AP cells, which you can see a little bit here. So AD to AP chain, and then they'll divide and become type B spermatogonia cells, which will then be developed into type B cells, and type B cells can further differentiate to become primary spermatocytes, which are going to undergo meiosis 1. All right, so once you have spermatogonial phase, we've gone from type AD to type B to our primary spermatocyte. Now you're officially in the spermatocyte phase, and so the primary spermatocyte is going to go through that meiosis 1. You're going to go from a 2N, 2 being the number of chromosomes, to just a single number to just N. And then you're going to have crossing over occur during prophase 1 of meiosis 1. And you may recall that we had prophase 1 actually had sort of subphases to it. There's the leptotene, there's zygotene, pachytene, and those 2 are where recombination crossing over is going to occur. Then diploatine and dikinesis overall. And so you can see crossing over occur at these chromosomes. All right, hopefully we won't get asked such a question, but going from 2N chromosomes to N chromosomes in meiosis 1. And here we can see, review of this. Right, our normal somatic cells in the human body, aka non-germ cells, are diploid, with 2N, N being the number of chromosomes, right? 23 pairs of chromosomes gives you 46 overall. Meiosis 1 is going to give you cells with 23 chromosomes, and then meiosis 2, you're going to end up with 23 overall. And so, yeah. All right, so that's stage 2, and then within stage 2, so we've gone from primary spermatocyte, now we're done with meiosis 1, we're going to have our secondary spermatocytes, and they're going to undergo meiosis 2 in just a few hours. Meiosis 1 is going to take about 2 weeks, and basically you're going to have, at the beginning, your secondary spermatocyte is going to have, one chromosome, which has basically 2 copies of the DNA for these, proteins, whatever it's going to make, which would be the chromatids, right? And then each spermatid is going to get, 1 version of each set of those, DNA, right? So if you have, a copy from your mom, a copy from your dad originally, now the end sperm is going to get a copy. This one's like, it's not the best analogy, but I think what I'm trying to say, which is like there's your spermatid is not going to have a second set of instructions, albeit a slightly different version of it, but a second set of instructions for the same coding, the same enzymes that it's eventually going to make. All right, so once you have your spermatotids, you're going to undergo spermatogenesis, where the spermatotids are going to go ahead and go from our early spermatotids, which are going to be circular shaped, and then they're going to end up in our late spermatotids, which are going to be what we typically consider a sperm to look like, right? And so spermiogenesis has 4 phases. The first is the Golgi phase, where you have the formation of an acrosomal vesicle from the Golgi apparatus. And this is going to contain hydrolytic enzymes. Then you have the start of the formation of the tail. After that, you have the cap phase, where you can see that the acrosome, again full of those hydrolytic enzymes, starts to flatten. The sperm tail is elongating, so it's a cap phase. Then you have the acrosome phase, which is the acrosome and nucleus condensed to form a pointed head. All right, so flattened acrosome, and we don't really see it as much anymore, but you can see we have the formation of a head. And then the maturation phase is we had the mitochondria are starting to move into the neck and forming a middle piece. And then our final phase is we're getting rid of the excess the excess cytoplasm, which we know our Sertoli cells will eat up from our histo lectures. All right, so that's not too bad. We know that the testes themselves, as we said, they're made up of these seminiferous tubules where all this is taking place. They're lined by these Sertoli cells, which are going to surround and nurture developing sperm, secreting growth factors that regulate mitosis and meiosis. They form the blood-testes barrier, which is also going to separate us into the adluminal and the basal compartments. It secretes fluid into the lumen of the tubules, and then secretes androgen-binding protein to maintain a high concentration of androgens overall. And then we know that the Leydig cells, aka the interstitial cells, are producing the testosterone in our testes. All right, and so those would be located, outside of our seminiferous tubules, those Leydig cells. All right, but just as a quick review, we had our spermatogonia as phase 1 of spermatogenesis. Then we had the spermicide phase, and then we have spermiogenesis, where we're undergoing the Golgi phase, cap phase, acrosome, and maturation to end up in our late spermatid that would look like what you'd consider a stereotypical sperm to be. All right, but remember at that point they still need to mature. All right, so more on the physiology of the testes. In terms of the Sertoli cells, we know that the first Sertoli cells are going to nurture the sperm, and so we know testosterone is really important. In particular, these Sertoli cells, in a way, kind of act like the granulosa cells from the ovary, even though we're talking about testes here. But it's going to the Sertoli cells are going to be stimulated by FSH, and then it'll start down this path with the G-coupled protein receptor. Adenylyl cyclase, protein kinase A will be activated as a result of FSH binding overall. Then you get, the new proteins will be synthesized. In particular, androgen binding protein or testosterone binding protein will make sure that testosterone is in high concentration for the sperm in the seminiferous tubule. All right, so follicle stimulating hormone, great. Basically the same pathway is happening inside of our ovary or granulosa cells of the ovaries. We also have the production of aromatase, which we know aromatase, just like in the granulosa cells, will convert testosterone to estradiol. All right, and then this is a two-part system, a two-cell system. So our Leydig cells out here are going to be stimulated by the luteinizing hormone. And so on the side of the female reproductive system, the Leydig cell for males is a lot like the theca interna cells for females, right? And so here we have luteinizing hormone is going to come in, bind to Leydig cell. We have a G-S coupled receptor. Adenylyl cyclase will be activated, protein kinase A, and then enzymes to convert cholesterol to testosterone. And then testosterone will go out into the blood through endocrine. That's what we call like an endocrine release, and then a paracrine to the adjacent Sertoli cells. All right, so not too bad. What about the actual creation of testosterone? Do we need to know the pathway? And I kind of feel like I hope not, but we know the broad strokes. So our Leydig cells are making this testosterone, and they need the luteinizing hormone to stimulate the uptake of ultimately, to produce the enzymes that they need for the conversion of cholesterol to testosterone. All right, so basically we have cholesterol coming into the cell through a side chain cleavage enzyme that is unnamed. We get the production of pregnenolone, and that will be converted by the smooth endoplasmic reticulum to progesterone. All right, that may sound familiar to you. That can then be converted into testosterone. Alternative pathway is the pregnenolone will be converted into dihydroepiandrosterone, DHEA overall, which can then be converted into testosterone. All right, and then importantly, testosterone can get converted into dihydrotestosterone by 5-alpha reductase. I'm pretty sure we'll have some sort of question on that because we know testosterone is going to do a lot of the development of internal male characteristics for development, and then dihydrotestosterone is more like development of the genitalia, external genitalia. All right, so moving on, we've already talked about this. The main function of the testes, endocrine, is the production of testosterone, and we have about 7 milligrams secreted daily by a young adult, and then spermatogenesis, and our testosterone is metabolized to make this more potent dihydrotestosterone, and it's done so by 5-alpha reductase, and 5-alpha reductase is found in the skin, prostate, and genital tissues, and yep, so that's pretty much that. Then we had activation of androgens in males. So one of the points that we have is dihydrotestosterone, DHT, binds more tightly to receptors, to the same receptor that testosterone binds to. And so basically, what are the actions of these two androgens? Just generally speaking, they're going to promote growth, differentiation, function of the accessory organs, which we know, of course, are like the seminal vesicles, the prostate, etc. It's required for spermatogenesis. Stimulate growth of chest, armpit, facial hair. Then we have, puberty. During puberty, to stimulate growth of secretions of sebaceous glands in the hair, grow the larynx, thicken the vocal cord, increase bone and muscle mass, increase food and appetite, increase libido. And then during embryology, we know it's testosterone for, as we said, the internal Wolffian ducts, formation of the internal genitalia, and then dihydrotestosterone, development of male external genitalia. All right, so a little bit more interesting, we've talked about, FSH is going to act on the Sertoli cells, just like FSH acts on the granulosa cells in women. All right, so follicle stimulating, you could think of like the seminiferous tubules are those like the follicles of the male system, and then the follicles for female, like granulosa cells are inside the follicles. So follicle stimulating hormone acts on the stuff that's in the follicles. All right, the male quote unquote follicle is the seminiferous tubule. All right, so then if follicle is something that eventually contains the cells that are the cells that are going to eventually become gametes. All right, so then how is this actually controlled? Well, we've talked about luteinizing hormone. Basically, we have the hypothalamic pituitary gonadal axis. We have luteinizing hormone and follicular stimulating hormone are going to be released by the pituitary gland, which you can see here, in response to gonadotropin releasing hormone, otherwise known as GnRH. And GnRH is produced by the hypothalamus, which you can see here, and it is released every 1 to 3 hours. And so this is, a release that is pulsatile, as we mentioned before, and it causes the release of luteinizing hormone and follicular stimulating hormone. All right, and then there's a point that was made. These are glycoproteins, and so what separates luteinizing hormone from follicular stimulating hormone, you have an alpha subunit and a beta subunit. The alpha subunit is going to be the same. The beta subunit is going to be slightly different. And so one of the things that's quite interesting is that if you recall, we said in our histo lecture, luteinizing hormone is responsible for causing ovulation and causing the corpus luteum to exist right after ovulation. But luteinizing hormone goes away after an initial spike for women, but the corpus luteum can be maintained by HCG from the placenta. And notice how HCG looks a lot like LH, right? The beta subunit is basically the same. Slightly different, but close enough that it can do the same job. All right, so then we had GnHR released by the hypothalamus. It goes to the anterior pituitary, causes FSH and LH to be released. We know FSH acts on the follicle cells, right? And for males, that means what's our quote-unquote follicle? The seminiferous tubule. And so it's going to act on our Sertoli cells there, cause the creation of cell products. We know one of those is androgen-binding protein or also called testosterone-binding protein, to bind to testosterone, make sure it's present for our developing sperm. We also have the production of inhibin. We also have the production of aromatase, which will take testosterone and convert it into estrogen. All right, so inhibin will go back and inhibit the release of FSH from the pituitary gland. Luteinizing hormone, we know, acts on the non-follicular cells. So for men, that's the Leydig cells. For female, that's the interna theca cells. And so our Leydig cells are going to take that. They're going to produce the testosterone, which will go to the Sertoli cells, or it will go to the blood. If it goes to the blood, it's going to do the secondary body effects, but it's also going to eventually, as a result, make it back to both the anterior pituitary, will act as a negative feedback loop and decrease the secretion of LH. And it will go back to the hypothalamus, where it will decrease secretion of GnRH. All right, so this feedback loop is going to be pretty important. GnRH is a pulsatile release every 1 to 3 hours, causes FSH to be released. FSH binds receptors on Sertoli cells, right? Sertoli cells are follicle cells in the same way that our granulosa cells are in women. All right, and then inhibin is amongst the products. It's going to inhibit the release of FSH. Androgen binding proteins among the products of the Sertoli cells. Luteinizing hormone binds to our Leydig cells. We already talked about all this. And then, yeah, this is basically just what we said. So we're going to go ahead and move on. Next page. So far, so good. All right, so what about these accessory glands? What do they do? We already, again, kind of talked about these, but the ducts are for transporting sperm, right? Eventually, we want the sperm to leave the penis. And we have the epididymis, which is important for sperm maturation. Why would that be the case? Well, we're leaving the seminiferous tubules without being able to move. So our sperm need to be able to know how to wiggle so they can move. And then our ductus deferens, important for sperm storage, but we also know that it's going to contract and be able to expel the sperm as well. Then we have the seminal vesicles, secrete fluid, nutrients, which fructose is a huge one. Prostaglandins for smooth muscle contraction, the Cowper's gland, which we see here for the mucus, prostate, watery secretions, buffers, right? If the vagina is acidic, you might want to have an alkalytic buffer in your semen, and then more nutrients. And then that's what this says here. All right, general concentration of sperm, greater than 100 million sperm per milliliter of semen. That's quite a lot. That's what you consider normal. All right, pathophysiology, so what can go wrong? Well, we know how important androgens are for the development of male external genitalia and internal male glands, ducts, etc. So what if you don't have, if you have a mutation to the androgen receptor? Well, you could have an individual that is XY in terms of their DNA, but be born as females because the androgen they produce is not being used appropriately to develop male characteristics. And so that's what's happened to this person. And this would be considered androgen insensitivity syndrome. There's 3 categories. We didn't really get too much information about this. So presumably this person right here is complete, but who knows? Another thing you could think about here is, well, if it's partial, then maybe, you still have like your female phenotypically, but genotypically you're male. And so phenotypically you're female, but genotypically you're male. So maybe some parts of the male internal structures did get developed and you have like a testicle, right? And then you're producing too much testosterone, and so you might have some clinical problems. All right, then we have aromatase deficiency. We know what aromatase does, testosterone to estrogen. We do have 3 different types of estrogen. Estradiol is the most normal, common one, estratriol, and that's for during pregnancy. Estrogen is required for fertility, maintenance bone density, fusion of the epiphysis. We were told in class when estrogen goes up with the onset of puberty, women tend to stop growing because it causes fusion of the long bones, and then resorption of fluid in the epididymis. So yeah, these are just some deficiencies of aromatase as a result of lowered estrogen. All right, so moving on. What if you didn't have DHAT as a result of a lack of 5-alpha reductase? And so DHT, we said, is responsible for external genitalia. So you're going to have ambiguous genitalia at birth, I mean externally, generally be assigned as girls. Wolffian ducts, so the internal structures will be normal. You'll have your epididymis, vas deferens, seminal vesicles, and then follicular stimulating hormone elevated, increased luteinizing hormone elevated, and presumably this is because if you have decreased presumably hypogonadism, then you don't have any feedback mechanisms to, say, lower this, so it's just going to continue to be increased. All right, which is similar in this case. So we have hypogonadism, so impaired testicular function from either basically primary testicular function. There's just something wrong with your testicles. And so we would say this is hypergonadotropic, which hyper means a lot. Gonadotropic means your gonadotropes are your FSH and LH, so high FSH and LH levels, but low testosterone levels, which would be the hypogonadism. Klinefelter syndrome is the most common. That's XXY genotypically. You could also have a poorly functioning hypothalamic pituitary axis, which would be hypogonadotropic, which means low FSH, LH, and then hypogonadism is low testosterone, and so this would HH, hypogonadotropic, hypogonadism, HH. Commonly, we have 1/3 cases is Kallmann syndrome, and this is anomosic. I can't say that, but you can't smell very well. Like you can't smell anything. Not that you're stinky. That's 1/3 of the cases. Basically, there's poor migration of the neurons associated with the release of gonadotropes, and I think they get stuck. For some reason, they start where the first cranial nerve is, and so they don't migrate. You can't smell very well as a result of cells being in the wrong place, I think. And then for men, hypogonadotropic, so low FSH, low LH, then you're gonna have low testosterone, erectile dysfunction, not enough sperm because you can't develop sperm if you don't have testosterone, and you can't develop it well. All right, we'll talk about what happens with females in just a minute. All right, so that was the male side of things. That took a while. Female stuff we're fairly familiar with because this is pretty much covered in the histo lecture, but we have the ovarian cycle and the endometrial cycle. I think this stuff is pretty interesting, but basically, what's going on with the follicles. We have development from primordial to Graafian follicle, then we have ovulation, then we have the corpus luteum taking over with the production of progesterone. We have our endometrial cycle, which is coinciding with this, which is preparing the uterus to receive an implanted blastocyst. So preparing for pregnancy. We have hormones involved here, estrogen, androgens, estrogens. Again, we're talking about women development, so breast development, fat distribution, bone distribution, uterus, liver, heart. Androgens are produced in the adrenal cortex. In addition to, well, we don't have testosterone here, but we do have the theca interna cells are making androgens. All right, so hormones in the reproductive cycle, just like with men, we have gonadotropin releasing hormone from the hypothalamus and causes anterior pituitary FSH and LH release, and then we have follicular stimulating hormone is going to work on the AK follicle cells, which is quite literally a follicle cell, and then it becomes a granulosa cell. Granulosa cells are going to produce estrogen under the influence of FSH. Again, for men, what are our quote-unquote follicle cells? They're the Sotoli cells, right? Our seminiferous tubule is like our follicle, whereas like for women's side of things, a follicle is a follicle. But what's inside the quote-unquote follicle? An oocyte, and for men, sperm, here oocyte is the egg, right? And then granulosa cell for women, and then Sertoli cell for men. All right, so we've talked about this in the histo lecture, so I don't want to go over too much, but this is the ovarian cycle again. First stage is the pulsatile release of FSH, follicular stimulating hormone. It causes our follicle cells to become granulosa cells, and those granulosa cells will be able to secrete estrogen. The granulosa cells take in FSH, take in testosterone, produce estrogen with aromatase. That estrogen is going to increase follicular growth, endometrial growth, and then part of the continued growth of the follicle, we're going to also have the theca interna will be created from the stroma around it, and so then the theca interna will take in cholesterol under the influence of LH and produce testosterone. All right, and so that's the first two steps. more granulosa cells, more estrogen being produced. Then you have a pulsatile release of luteinizing hormone from the anterior pituitary, and this pulsatile release is going to cause the granulosa cells to produce, it's going to have a negative feedback on the granulosa cells, produce less estrogen, but also cause ovulation. So some of the granulosa cells are going to leave as the corona radiata, but most of them are going to stay back with the theca interna cells and become the corpus luteum. And when they stay back, they go from theca interna to theca lutein and granulosa to granulosa lutein cells, and they're going to shift gears, and now they're going to mostly produce progesterone, right? Pro-gestation, pro-pregnancy, right? And so now we had, lots more granulosa cells, more estrogen being produced. Now we have ovulation, less estrogen, and then a shift to granulosa cells are still going to produce some estrogen, right? We see estrogen is right there, but it's going to mostly produce progesterone, which is why this is higher up right there. Okay, and then we said, the corpus lutein is producing this stuff. Corpus lutein was created as a result of LH, so it's dependent on LH, but we see that LH goes away. So this guy's going to die if there's not a replacement for LH. What was the replacement for LH? We said that was right here, if this will load. HCG, which comes from a placenta, right? LH looks like this, HCG looks like this. All right, so that was that. We had then, as hyperthalamic pituitary axis, GnH is released in pulsatile fashion, causes FSH LH, LH binds to theca interna, causes biosynthesis of testosterone and progestins, which would include progesterone. Androgens enter the granulosa cells, get converted to estrogens. So that's this thing right here. We have LH on our theca interna cells, helps produce the enzymes we need for the conversion of cholesterol into testosterone, which is then released into both the blood, and then also goes through paracrine distribution to our granulosa cells, which then becomes estrogen, as our granulosa cells are stimulated by follicular stimulating hormone. All right, so not too bad. FSH binds to granulosa cells to increase synthesis of aromatase, activins, inhibins. So this is all basically the same as what's going on in males. So here we have our overall distribution: hypothalamus releasing gonadotropin releasing hormone to the anterior pituitary that causes the release of FSH, which goes to our granulosa cells. That FSH does the G-coupled protein receptor pathway, produces aromatase, which is going to allow for the androgens that theca interna cells release to be converted into estrogen. That estrogen is going to go back and inhibit the anterior pituitary as well as the hypothalamus. And our granulosa cells will also produce inhibin, which will do exactly that, inhibit the anterior pituitary. And then they can also produce activin to activate the anterior pituitary. All right, progesterones, aka progesterones, aka progestin, will work on developing the endometrium to prepare for implantation of a blastocyst. All right, LH we know will go to theca interna, stimulate the release of androgens from the conversion of cholesterol to androgens. Progesterone would be released, and we got the story. So the question is, what's going on with this, with the positive and negative here? Because estrogens and androgens are typically, this is a negative feedback situation, but we actually did something a little bit more complicated, just annoying, but also kind of cool. All right, so before we get there, we have during puberty, early in the follicular stage, gonadotropes and the pituitary are not very gonadotropin releasing hormone sensitive, and so we have a pulsatile release, but it's going to give you a small surge in LH, right? And so we have our pulsatile release every 1 to 3 hours, and so at the beginning of the follicular phase, we don't have a large sensitivity to gonadotropin releasing hormone, and so only really FSH gets released by the anterior pituitary. But then during late follicular stage, the estrogen that's being released by the granulosa cells is going to cause there to be a positive feedback during this 2-day window right here, and it's going to cause a positive feedback, make the anterior pituitary more sensitive to gonadotropin releasing hormone. Oh my goodness. And as a result, you will get a surge in LH. All right, so that's what this little graph is saying overall. All right, so what if we have some problems? Because there's always problems. We're back to our Kalman syndrome. Basically, women use GnRH just like men, so you could have Kalman for women. And, disordered migration of the GnRH hypothalamic cells during embryo development. Kalman's is HH, which is hypogonadotropic hypogonadism, which means low FSH, low LH, low estrogen for women. All right, and so for women, that means estrogen's involved in breast growth, so poor breast growth, and then you're not going to have the development of the endometrium very much, and therefore you're not going to have periods as much. All right, next problem that can go wrong is endometriosis. So basically you have endometrial tissue, right? The lining of the endometrium of the uterus is showing up where it should not. And the endometrium is, it responds to estrogen, right? Because it grows in response to estrogen to prepare for implantation in the normal situation. So if you have if endometriosis, this isn't stuff that's not supposed to be, or is in the wrong spot, that responds to estrogen, well, you're going to have, pain when these start to grow, and it's just not very good. So how can you do this? Just make sure you have less estrogen overall, which means less GnRH or less GnRH receptors. Okay, so here we have more on the hormonal control. So early to mid follicular phase, right, as our follicles developing, low levels of estrogen exert a negative feedback on GnRH, FSH, LH. Estrogen promotes more estrogen secretion by the follicle. Okay, so all right, estrogen is going to have a negative feedback overall. Then late follicular phase, estrogen is going to have a positive feedback to make us more sensitive to luteinizing hormone in particular. All right, and then we're back to negative feedback overall. All right, so activins and inhibins. Inhibins suppress FSH production. Activin enhances it. Not too much to know there. Feedback, we already talked about this. So I think we're good. All right, creation of steroids. We went over this. 2-cell model, we went over that. Summary of the hormones and the functions. Let's just talk quickly about relaxin. So inhibits contraction of uterine smooth muscle during labor, relaxes pubic symphysis. Inhibin inhibits release of FSH and to a lesser extent LH. All right, so that's it. Good job, guys, and we keep at it. It's pretty late right now, but we're some awesome people who try pretty hard. So, all right, see you next time.